Okay, so thank you everyone for being here. I just want to say I appreciate everyone sticking through to the last talk of the final symposium section. My name is McCloskey. I'm a PhD student at the University of Rochester. I work under Dr. James McGrath, and I'm excited to present my project on developing a reproducible human blood-brain barrier model for in vitro studies of cognitive disorders. Just gonna grab the laser really quick. Okay. So this project was really motivated by a need for a human model to study sepsis, which is a life-threatening organ dysfunction caused by a dysregulated host response to infection. So essentially, factors from a local infection enter the bloodstream, and then they'll go throughout the body and cause inflammatory responses that can actually cause damage to your vital organs. As such, it's the leading cause of death in the ICU, as well as the most expensive condition to treat. Interestingly, approximately 1/3 of sepsis survivors develop cognitive dysfunction, and while the mechanisms of this brain injury are really complex, there is evidence that suggests that the blood-brain barrier plays a role in some of this dysfunction. Everyone's probably seen some talks on the blood-brain barrier, but just a brief introduction. The vasculature within the brain is unique compared to the periphery in that it's highly selective and semi-permeable. The endothelial cells that make up this barrier are characterized by tight junctions and very low permeability, so it's really regulating what can go into and out of the brain. The region that we're particularly interested in our lab is this area called the postcapillary venule. It follows our capillaries, and during inflammation, we get the presence of this perivascular space. So if we look at our inset, we actually have this separation of one of the support cells called astrocytes from our endothelial cells and our parasites. And why we're interested in this is because this is where most of the immune cell migration takes place, which is really important when we're thinking about the brain damage that happens during sepsis. So one thing that is really interesting for our lab, what we found pretty striking was that some people, there's a population that seem to have potentially underlying cognitive pathology, but they've been resilient to any sort of symptoms, who get sepsis, and following the systemic inflammation, they emerge with what appears like a late set symptomology. So if you're thinking about Alzheimer's disease, there might be some pathology, but no one knows that it's there because they're not symptomatic, and this single inflammatory event basically progresses the disease to really late stage. And so a common thread between both sepsis and Alzheimer's disease is breakdown of this blood-brain barrier. So in thinking about modeling the blood-brain barrier in Alzheimer's in our project, ApoE4 being the greatest genetic risk factor for developing Alzheimer's, as well as already established links to blood-brain barrier dysfunction, emerged as a promising candidate. So if we look at our inset here, ApoE is secreted primarily by astrocytes within the brain. The most common allele that we'll see in the population is this ApoE3 allele. When secreted by the astrocytes, it can bind onto a molecule on our parasites, which protects the blood-brain barrier from degradation. However, the ApoE4 allele seems to struggle to bind to this protein, and this ends up activating a pathway that upregulates MMP9, which leads to basement membrane degradation, tight junction degradation, as well as ultimately breakdown of this blood-brain barrier. So my goal is to develop a microsim human blood-brain barrier model for this reductionist approach to elucidate the mechanisms that might lead to the enhanced vulnerability of the ApoE4 population to brain damage during sepsis. So just to introduce our microsim platform, it is a microphysiological system enabled by a silicon nitride nanomembrane. These membranes are ultra thin, highly porous, and optically transparent, and this is just an example of a membrane here that we've put within a handmade device. So we used to hand build all of the devices in the lab, and as people can imagine, this had a lot of challenges. So in particular, it was really difficult to scale up as our collaborations grew. This was high demand, and it wasn't necessarily always reproducible. It took hours of assembly to make just a handful of devices, as well as requiring expertise and training. So our solution was to develop what is called the modular microsim. So we worked in conjunction with a company called A-Line, and what we designed was an ability for people to actually make their own devices, whether or not you have an engineering background. So we utilize these fixtures shown in gray that combine our components and our membrane chip together. So this is component 1 shown in red. This is our bottom channel component 2 with the green and the blue. And then our membrane chip is shown here, and it uses pressure-sensitive adhesive to irreversibly bond these pieces together. And this is just a picture of what the final device looks like. This is a standard microsim that's been optimized for ease of culture. So we have what we consider like a transwell style format with an open well for cell culture on the top. And then we have these access ports that fit your standard pipette tips so that you can access the bottom channel. The membrane chip has this optically transparent portion and porous region that comes in sort of different flavors, I guess, so different styles depending on your particular need. But everything that I'll be showing in this presentation uses our nanoporous membrane. So this is approximately 100 nanometers thin. The porosity is near 15%, and the pore size is an average of 50 nanometers in diameter. So initially with this device, we wanted to establish that we could develop a reproducible blood-brain barrier model with one of our collaborating laboratories. So in order to show this reproducibility, what we did was we sent them assembly fixtures, components, and membranes, and then we also gave them these instructional steps and videos. So these were collaborators at the University of Bern, so they're very physically distinct from the University of Rochester, and they're really a biology and immunology lab, so not an engineering lab. And then side by side, our labs developed or used this EECM BMEC-like cell differentiation protocol that establishes brain microvascular endothelial-like cells that are really optimized for looking at immune cell transmigration in response to inflammation. And then separately, we made our devices, cultured cells, and then compared the results that we got between the different laboratories. So starting with the work that I did at the University of Rochester, this is just an example of a phase image that you can get of the cell culture grown within this well just by putting your device under a standard microscope, so there isn't any sort of finagling that's needed to visualize your cells within our devices. And then just to confirm that we have good maturation of our barrier, we stain for junctional molecules such as adherence and tight junction molecules, and we see nice expression of those, as well as response to inflammatory stimuli. So we're looking at cell adhesion molecules such as ICAM1 and VCAM1, and then we see this constitutive expression of ICAM2. So then looking at the University of Bern's data, just note there's a difference in scale, but we do still see good expression of our different adhesion molecules as well as our junctional molecules and response to inflammation. But in order to look at this a little bit more closely, what we did was we went ahead and quantified the upregulation of ICAM1 in the two labs. And these are taken on two different scopes, but what we see is that in both cases, in response to our inflammatory stimuli, we get approximately a twofold upregulation of the ICAM1 compared to our non-stimulated condition, and we don't see any significant differences in the data between the two labs. So thinking about developing a blood-brain barrier model, assessing the barrier properties of this is critically important. So what we did was we developed a sampling-based permeability assay. That's similar to what you would do in a transwell. So you add dye into the top well, you give it an hour to diffuse across your endothelial cell barrier and your membrane and into the bottom channel. And what we needed to optimize was a way to then sample the volume of the channel reproducibly while getting the entire sample of your dye. So what we developed was this method where you take a reservoir tip and you insert this with just blank media into one port, and then you'll take a separate empty tip and you'll actually reverse pipette out the volume from the channel, as you'll see here. And what you can do with that sample is just add it into a standard plate and then read the fluorescence. So this is the reverse pipetting technique. And what it allows us to do is then use just the standard calculations to see what our endothelial cell permeability is. In order to validate that this was a sufficient process to fully clear the dye from the channel, we did COMSOL modeling to show the dye going into the channel over a course of an hour. We're using lucifer yellow, which is a pretty small tracer molecule, which we need if we're looking at something like the blood-brain barrier. That's really tight. And then this is just modeling the sampling process. And what we see is that a reservoir of 50 microliters is sufficient to clear almost 100% of the dye, and we validated this experimentally as well. So using this assay, our two labs cultured cells in the modular microsim for 2, 4, and 6 days, and we also did a culture in the Transwell system just to show that what we're getting in our device is comparable to what you see in Transwells. And we get similar maturation of the barrier over time, and by our 6-day culture between the two labs, we see no differences in the barrier formation, as well as no difference between what we get in a 6-day Transwell culture. So with this, we feel like we've established well that we can produce reproducible results between two labs with this relatively complex iPSC-based blood-brain barrier model. So then what I wanted to do was develop a model that was more physiologically relevant. So now I am incorporating not just the endothelial cells, but also iPSC-derived brain parasite-like cells. So now we add our brain parasite-like cells into our bottom channel. When we seed them, we actually flip the device over so that they will adhere to the top surface of the channel, and then our endothelial cells are still cultured in the well. This is just a phase image of what that co-culture looks like, so you can visually confirm that you have both of your cell types, and you can also optimize the confluence, like the seeding density of your parasites to get the confluency that you want. The ratio of ECs to parasites in the brain is higher than most of the vasculature, so we're aiming usually for 70 to 90% coverage. And then in our porous region, you can see here with this setup that the two cell types are really only separated by this nanoporous membrane. And that's illustrated a little bit better in this video. So we have our endothelial cells stained for VE cadherin in the cyan color, and then our parasites are stained for PDGFR beta in this magenta. And as this video rotates, you'll notice that we don't actually see any separation of these two cell types, even though they are separated by our nanomembrane. So then as we look here, we start at the top of our endothelial cells, and it transitions down into the parasite layer. There's this really smooth transition of the two cells, so we really think that we have an opportunity to have a co-culture model that doesn't have this sort of membrane resistance to paracrine signaling. So using this model, we wanted to test how it was responsive to septic-like stimuli. We used a concoction called Cytomix, which is an equimolar mixture of cytokines TNF-alpha, IL-1 beta, and interferon gamma, which all seem to be upregulated in sepsis. And we see a dose-dependent response of both our monoculture and co-culture setup. What was interesting to us is we could kind of characterize these responses into different categories. So if we think about a tight barrier, you'll have a confluent monolayer, the junctions are matured, and the permeability is at or below our threshold of 0.6 times 10 to the minus 3 centimeters per minute. Then if you have a lower level stimulation, you can enter sometimes into this leaky range. So you'll get on occasion this very small gap in your monolayer that we can find under our microscope, or sometimes it'll be a confluent monolayer, but there's been loosening of the junctions, so it will become leaky. But with higher stimulation, sometimes we'll find that we get medium to large gaps in the monolayer that, again, we can visualize here. This isn't necessarily correspondent with cell death, but it is a high response to a cytokine stimulation. And again, we can visualize this on our layers before we do our permeability assay and before any need for any staining. And then finally, just to introduce some data that I have recently acquired that is working on developing this Alzheimer's model, cells from a sporadic Alzheimer's disease patient who is homozygous for ApoE4, and differentiated these cells into endothelial, our EEC and BMEC-like cells. And what we can pick here as we compare it to a healthy control is there's already some differences in just the morphology under phase microscopy. So it looks like we've got these string-like structures throughout our cell monolayer. And then we've got more jagged junctions, so this is staining for claudin 5, which is one of the tight junction molecules, as well as ZO1. And there also appears to be more ZO1 located within the nuclei as opposed to just at the junction. So this is indicative of potentially some barriers that aren't maturing as nicely over our 6-day culture. And what we find is this then corresponds to leaky or baseline barrier properties. So using our permeability assay, we looked at how these cells did in terms of letting lucifer yellow diffuse across. And we see that the cells that were derived from our Alzheimer's model were more permissive to lucifer yellow passing across this barrier, as well as more that entered really our leaky range as opposed to our light range or tight range. So with this, I'd just like to conclude and say that what I've demonstrated today is that our modular microsim platform has the potential to develop reproducible blood-brain barrier models. We really showed this between two physically distinct laboratories with different expertise, getting very similar culture results, even though we were using a complex iPSC protocol. We further went on to develop a microsim human blood-brain barrier model that now incorporates parasites, which are critical if you're thinking about communicating with endothelial cells and basement membrane formation. And then just a little bit of work developing an Alzheimer's model that already is showing potentially that these endothelial cells have intrinsic weaknesses in barrier properties. So future work would go on to establish really how the healthy control and our Alzheimer's models are different in their response to septic-like stimuli, particularly evaluating immune cell migration, which we can visualize on our platform, and then evaluating whether or not healthy control parasites could maybe rescue some of the weaknesses that we see in our Alzheimer's model. So with that, I would like to thank everyone for being here. I would like to acknowledge the support from my lab, as well as my mentor, James McGrath, family and friends, funding sources, and then finally, I will be looking for a postdoc at the end of the year, so if my stuff interests you, feel free to look me up and reach out. And with that, I will take any questions.
So thank you, Molly. We have already one.
Very nice talk. I was wondering, how is your plan to discern or discriminate between general variability? Because you see, you have one patient line, right?
Sorry, can you say that one more time?
You have one patient line only, right?
Oh, yes.
So
yeah.
how you will discriminate between, different polymorphisms from like different patients and the disease in the future?
Yeah, so future work will use other clones. So we have, I guess originally our plan was to use an isogenic line that converted just the ApoE3 to ApoE4 so that we could really pick a point this difference, but we had some issues obtaining cells for a variety of reasons. So right now we're looking at a different patient and a different healthy control, which does complicate the study, but working with a group of people, we would hope to obtain obviously biological replicates from different patients and go from there.
Very nice talk. I'm just wondering, why don't you select the FAD patients samples for reprogramming?
Because why it's sporadic Alzheimer's?
Yeah. FAD is gene related. It will display more significant phenotype. But SAD is aging related. Normally, we cannot find the significant phenotype for the IPIDY of the neurons.
Yeah, so this again goes back to our issue in obtaining the ApoE4 cells that were converted. So this was a cell line that became available to us, and we wanted to really get going on this project. So there are definitely some complications, and we would love to probe more into the different types of Alzheimer's disease, but this is the particular cell line that we happen to have on hand now. And so we'll learn what we can from here and then go on. Yeah, thank you.
Okay, thank you. Thank you for being the last speaker.
My pleasure.